the days of living in Townsville. All roads lead back to Townsville. But Katie has moved on to bigger and better things. And today she is, even though we were at the Coral Reef Center of Excellence for Coral Reef Studies together, Katie is not talking about anything related to coral reefs. She's moved on to the land and is talking about something that I think will be very relevant to everyone who's been working in the Threatened Species Hub and just sort of conservation decision-making in general, about translocation of species and trying to sort of understand these different management interventions that we are beginning to put in place as ecosystems deteriorate, and we're needing to be a bit more innovative. Katie is now a postdoc at the National Socio-Environmental Synthesis Center in Maryland, my home state. So are you actually based in Maryland now, Katie?
I'm actually in Boulder, Colorado right now. My collaborating mentor, Laura D, is based at CU Boulder. So I'm hanging out here for a few months.
Lovely.
Yeah.
I guess one of the bright sides maybe of a global pandemic is that we don't necessarily need to be in a particular place. So very excited to hear from Katie. Yes, so take it away.
Thank you so much, Amelia. So, share my screen. All right, are you guys seeing the title slide? Yes, no?
Yep.
Okay, great. Okay, so thank you guys so much for having me here today. As I mentioned earlier at Morning Tea, I'm talking about some work from my PhD while I was a student at JCU as part of the ARC COE, but this was work that I did while I was a visiting scholar at QUT. So it was some work looking at using ensemble modeling for conservation. So first, I want to acknowledge my colleagues that have been directly involved in this work. Mike Bode was one of my PhD supervisors and my main collaborator in this work. The Applied Mathematical Ecology group at QUT gave me some really great feedback while I was working on this research. And I'd like to especially point out Kylan and Chris, who are collaborators on a project that I'll discuss later in the talk. Megan, Leslie, Saul, and Colleen are also fantastic collaborators involved in that second project. And finally, I'd like to acknowledge the Yagara, who are the traditional owners on the land on which QUT is situated, where I did most of this research. I'd also like to acknowledge the Cheyenne and Arapaho peoples, the traditional custodians of the land on which I'm speaking to you from today, from Boulder. So the first part of my talk is on my paper with Mike on ensemble modeling and assisted migration, published last June. I'll talk about what assisted migration is and why it's controversial, what ensemble modeling is, and how we used it in this project. Then I'll talk about an extension using the same methods on an actual conservation project that's currently underway on Dirk Hartog Island. Our team of mathematicians and ecologists modeled various management strategies for one of the largest refaunation projects to date. So many species face extinction as a consequence of habitat and environmental changes they can't escape. Assisted migration is a conservation initiative that moves a portion of a threatened population beyond its native range when species are unable to expand or shift their range on their own. Translocation is used to describe the action of moving individuals from their native population. Assisted migration is usually proposed as a way to prevent extinction due to the effects of climate change, where species are sessile or there's some sort of barrier to prevent a shift to a more optimal thermal habitat. Assisted migration of the Edith's checker spot butterfly to prevent its extinction has been fiercely debated since the 1990s. So this butterfly was sort of the poster child for assisted migration. In 1996, Professor Camille Parmesan famously documented that the southern populations of the checker spot butterfly were extremely vulnerable to local extinction due to climate change. And some years after publishing this work, she advocated for the use of assisted migration to preserve some of these populations, which is really what got assisted migration in the public discourse. The Kino subspecies is indigenous to Southern California and Northern Mexico, and its overall range was diminished due to a suite of threats like urban and agricultural development. Its remaining intact habitat is at risk of becoming unsuitable due to warming and drying from climate change. It's been unable to expand its range because it can't navigate around the sprawling urban area of Los Angeles. So it would need human intervention to translocate cocoons to suitable habitat north of LA. So advocates of assisted migration insist that this is the only way to save species that are unable to expand their range on their own as climate change continues to worsen. Assisted migration has also been invoked to restore ecological functions and protect species from other threats. This is a burrowing bettong or a booty, a macropod that is being translocated to an island refuge in Western Australia. This translocation will not only protect it from non-native predators like foxes and feral cats, but the booty will increase soil turnover on the island, which makes it easier for other species to create their own burrows. Assisted migration is extremely controversial. It's been called ecological roulette, as we have a history of moving species outside of their range for some purpose, and those species subsequently wreak havoc in their new homes. This is well documented in the literature on invasive and pest species. So we have an ethical quandary. Do we save one species and potentially harm the extant species in the recipient ecosystem? Ecological communities are essentially a type of network. Here, each blue node represents a species and the lines represent pairwise interactions. Assisted migration has been pretty well studied in terms of what happens to the focal species represented by the green node here. What's not well studied, however, is how the translocation affects the species that are already present. If we introduce a new species, do we destabilize that network and cause the species that are present to become locally extinct? There are three general outcomes following assisted migration. In the best outcome, the species is translocated and it becomes established and it doesn't harm any other species. In a failed outcome, the species is translocated, but it doesn't successfully establish in its new habitat, but it hasn't negatively impacted other species to the extent that they become locally extinct. Although the translocated species may have failed to establish in its new habitat, it may have persisted in its native habitat. This depends on how threatened that species is and if removing individuals from the population in the native habitat negatively affects that population's chance of persisting. This depends on factors like abundance and genetic fitness and other factors as well. So this is the worst outcome. After a species is translocated, it somehow impacts the recipient ecosystem to the extent that some species that are there become locally extinct. Given the possible outcomes, we wanted to see how often each of these occurred. Then we wanted to see how many species were lost when things went really wrong with assisted migration. We were also interested to see if these outcomes held in different contexts. One of those that I'll discuss today is how the density of interactions within networks impacts the outcomes of assisted migration. The nodes and networks can be sensitive to the network structure, especially the degree of connectance in that network. So if species are more interconnected, there's a chance that the introduction of a new species will directly or indirectly impact other species. So to answer these questions and gain some general insight into the effects of assisted migration, we set up an ensemble of theoretical ecosystems. Ensemble modeling is essentially running the same general model over and over, thousands of times. It lets one change the model slightly, either through small changes in the model structure or parameter values, and then compare the output. The ecosystems we simulated were comprised of 15 coexisting interacting species using Lotka-Volterra models, which is the equation shown here. It's a classic equation used to represent interacting species. I'm sure most of you guys have seen it. So the change in abundance of species I through time t depends on the intrinsic growth rate and the abundance of that species. In our models, the intrinsic growth rates for each species were randomly chosen from a uniform distribution. The interaction matrix is comprised of elements X, which represent the per capita interaction strength of species J on individuals of species I. These per capita values combined with the abundances of species J and species I gives the amount of interspecific competition from species J onto I and the amount of intraspecific competition within species I. So we changed the interaction matrices for each simulation by changing the degree of connectance between species randomly and used the magnitude of, and randomly changed the magnitude of the interaction using random parameter values. To generate random levels of connectance, we used a Bernoulli random variable to decide if species were connected or not. So where species did not interact, there was a 0 in the interaction matrix. This let the degree of connectance randomly vary across our models in the ensemble. We then used another Bernoulli random variable to decide if the non-zero interactions were positive or negative. And then we fixed the diagonals to always be negative to ensure density dependence from within species competition. We then used a uniform distribution to allocate the values of the parameters. So these were run to equilibria, and any models where the 15 species did not coexist were discarded. It was really difficult to find models of 15 species with stable coexistence. So out of the millions of simulations we ran, we kept 100 ecosystems. So even though we didn't necessarily parameterize the models to represent any particular ecosystem, by only keeping models with stable coexistence, that allowed us to discard models where the parameters were not ecologically or biologically feasible. So we wouldn't have had stable coexistence of 15 species where the parameters were really high or really low. So we had the 100 coexisting ecosystems. And then once the 100 ecosystems were found, we simulated assisted migration. A single randomly generated species was added to each ecosystem, and the response of the ecosystem was monitored for 100 time steps. So we wanted to see, did we end up with the best case scenario with 16 species in the new ecosystems? We repeated the whole process 100 times for each of the ecosystems with a new randomly generated translocated species each time for 10,000 simulated assisted migrations. What we found was that the best outcome, where the translocated species became established but didn't negatively impact its new community, occurred 61% of the time. Failed translocations occurred 17% of the time. And the worst outcome, where species within the recipient ecosystem became extinct, occurred 22% of the time. So using the results from our ensemble model and a risk analysis, we calculated the average total species richness when assisted migration is not chosen and when it is. The graph here shows the probability of the translocated species in green persisting in its native habitat at 50%. So we set this to represent a critically endangered species by IUCN criteria. This persistence of this species increases to 90% when you included translocated species that successfully established in the recipient ecosystem. This leaves a 10% chance that species will not persist in any outcome when assisted migration is or isn't chosen. This blue distribution is the possible numbers of species within the recipient community persisting. So 80% of the time we have 15 species within the recipient community. So that represents the best and failed outcomes where the translocated species doesn't negatively affect the recipient community. When the recipient community is affected, there's a rare but still possible chance of losing multiple species within the recipient ecosystem, bringing the species richness down to 7 species in some instances. So if one does not proceed with assisted migration, the total species richness is 15.5. This is, the 0.5 is a 50% chance that the translocated species survives on its own. And the 15 is due to the fact that the recipient ecosystem wouldn't be affected because you don't choose to translocate anything into it. So if one does proceed with assisted migration, the total richness is slightly lower with an average of 15.4 species. In the best outcome, you have 16 total species, which has our 15 recipient ecosystem species and the translocated species. But this is offset by the instances where multiple species are lost. So in our baseline scenario, we allowed density of interactions to vary across models in the ensemble. We also modeled scenarios with 3 specific levels of network density to see if our results were sensitive to that. The low density network allowed for 25% of the interactions to be non-zero. The medium density network had non-zero values for 50% of the interactions, and the high density network had non-zero values for 75% of the interactions. So our results were the same as a baseline scenario if you don't choose assisted migration, because we didn't change IUCN criteria or anything like that. But if you do choose assisted migration, the overall species richness is worse, and that was especially pronounced in models with moderate or high densities of interactions. So to sum up, assisted migration is risky, and in an unparameterized system where the only concern is total species richness, assisted migration can cause more harm than good. The outcomes will vary based on the attributes of both the recipient ecosystem and the translocated species. We didn't explicitly model trophic dynamics or specify interaction type, but that would impact the outcomes. We used a very simple utility function in our analysis where we assigned equal weight to the translocated species and to each species in the recipient ecosystem. So this is unlikely to be realistic for a real project. Usually a higher value would be associated with certain species, particularly the translocated ones. So therefore, it's important to consider the conservation status as well as the roles and functions of affected species when determining if assisted migration is worth the risk. So in some cases, even a small change in the population might be an unacceptable outcome. So assisted migration is a viable conservation intervention, but the risks have to be offset by either reducing the impact of the translocated species on the recipient ecosystem, or you have to ensure that the translocated species persists even if it becomes extinct in its native habitat. So we took this ensemble modeling framework and extended it to an actual conservation project currently being implemented by the Department of Parks and Wildlife in Western Australia. Dirk Hartog Island is located in the Shark Bay World Heritage Area off the coast of Western Australia. And as I mentioned at the beginning of my talk, this is one of the largest refaunation projects to date. The goal is to restore the island's ecosystem to how it was before European settlement. Goats, sheep, and feral cat had to be eradicated, and 13 native animals, including some mammals, marsupials, and a bird, are being translocated to the island at different times. The biologists in WA have translocated a few of these species so far, and one of the first is the banded hair wallaby pictured here. These 13 species were thought to be present on the island before human interference. Goats and sheep impacted the vegetation, reducing the food source for many of the species. And feral cats were effective predators of the rodents and small mammals and competed with the carnivorous mammals. So because these 13 species would be translocated over a short time frame before 2030, the biologists in WA needed to see if priority effects would change the overall outcomes. Often translocations of multiple species are temporally spaced out, and most projects only move a few species. 13 different species in this short of a time span is unprecedented. We also wanted to see if some species were more sensitive than others to particular reintroduction strategies. Was there one strategy that worked for all species equally well, or were there any species that did poorly no matter what? So though the 13 species were thought to have coexisted on Hartog Island before European settlement, they have not been directly observed together. Since they're a novel assemblage, there's a lot of uncertainty in how the species will interact. So my colleagues and I at QUT work directly with the biologists at DPAW and WA to incorporate data into a model ensemble. 3 biologists gave us a total of 6 sign-structured interaction matrices that they made independently, and a consensus matrix that they developed after discussing together the possible interactions in detail. These matrices represent different dynamics that may occur when the species have all been translocated to the island. They are the aggregate of a variety of interactions like predation, competition for food or habitat, as well as some positive facilitative interactions. These included the interactions between the 13 key species and interactions with functional groups that represent species already on the island, like vegetation, other rodents, predatory birds, and varanids that can be prey for some of the translocated species and predators of others. We generated hundreds of thousands of interaction matrices with random parameter values with the appropriate sign and matrix structure. So the reintroduction strategies given by the experts were 23 different scenarios that specified where and when each species would be translocated onto the island. Generally, the locations were fairly consistent due to habitat preferences, but timing and order differed. And the locations were in part due to habitat preferences of each species and also the ease of translocating species. So there are 4 different sites on the island that they could be translocated to. The locations of the translocations and the dispersal data informed the spatial component of the model, and species had different dispersal rates, and there was a time lag for most species before they started dispersing. We used certain criteria to discard models that were ecologically unviable, which we called model filters. These included the coexistence of subsets of these species due to their co-occurrence on other islands and within the fossil record. We also used the upper and lower bounds of recorded growth rates of these species as another filter. So models where any of the species reproduced outside of this range of feasible and biologically realistic growth rate values were discarded. So we followed a similar methodology as the assisted migration paper, but this was parameterized for the system as closely as possible, given the amount of uncertainty in the system. So here we have the aggregate results from the consensus sign structured matrix, which is the one that the biologists came together and worked on. This is for all species across the 23 reintroduction strategies. The black dot in the violin plots is the average number of species lost by 2060, which is roughly 30 years after the last translocation. On average, there's about 1 failed translocation for each reintroduction strategy. Option 6 is slightly worse than some of the other reintroduction strategies. And this is the option where the booty burrowing bettong is not translocated. This is probably because some of the interaction matrices emphasize that species eco-engineer role, where it helps facilitate other species to make their own burrows by increasing soil turnover. So by not translocating it, that function is lost, as well as some of the positive impacts on other species. For all of the reintroduction strategies, there's a very small chance of losing multiple species. The tails of the violin plots extend to about 4 failed translocations. So this heat map takes those same results, but separates it out by species. The gray is where the failed translocations are effectively 0, which is great. The cool colors represent possible translocation failures, but a low probability. And the warmer colors represent higher instances of failed translocations. The outcomes are fairly consistent across the reintroduction strategy for most species. So on the far left, we have the dibbler, which doesn't do very well no matter what reintroduction strategy is chosen. There are some exceptions though. There's a fair amount of variability with how the heath mouse fares across the reintroduction strategies. This heat map takes a species-specific response to the reintroduction strategies and breaks it down by the sign-structured matrices developed by the biologists. So each column represents a different sign-structured matrix. This is only for a subset of the species to show the different kinds of outcomes, but we have 13 of these. By separating the results out by the interaction matrix, we can see that some species are more sensitive to the reintroduction strategies, depending on what dynamics are thought to be occurring. So the dibbler fares fairly well in some of these strategies. You can see that there are some gray columns, and it does better in the left 6 columns than the yellow 7th column, which was the consensus matrix developed by the biologists. So other species like the chuditch or the booty seem to do well no matter what. They're fairly insensitive no matter what reintroduction strategy is chosen and no matter what sign-structured matrix is underlying the dynamics. So the other species vary a bit. So we have the mulgara and the greater stick-nest rat here, and there's some variability across interaction matrices and reintroduction strategies. And then finally, the heath mouse just has a lot of variability, no matter what interaction matrix is chosen or the reintroduction strategy. So in both examples, even though we use very simple models, the differences in the model structure through interaction matrices and parameter values yielded quite a bit of variability in the results. This underscores the importance of getting the model structure as accurate as possible within reason. Accuracy in parameter values is also helpful, but without having data from mesocosm experiments or observations, parameter values for interactions between species are rough estimates at best. In attempting to represent any complex ecosystem with models, there will be significant amounts of uncertainty. It's not feasible to include every biological and ecologically relevant parameter for each species. Using ensemble modeling with relatively simple models allows one to capture the necessary dynamics and allows for the flexibility in parameter values where there's uncertainty. This also allows one to evaluate multiple scenarios. Even better, the model ensemble can be updated with more information as the data are gathered to further constrain outputs. So this can facilitate adaptive management and conservation decision-making by taking some management options off the table and allowing for the evaluation of newly developed management options post hoc. Finally, this method helps us to quantitatively evaluate the aggregate and species-specific responses to different conservation initiatives. This allows us to consider if outcomes and risks are worth having to save a particular species using quantitative predictions and not just discussing them qualitatively. So thank you for your time today. The first paper I discussed is published in Conservation Biology, and the other will hopefully be out soon. We just submitted it last week for review.